The blood-brain barrier, it's not a structure that you can like put your finger on or like a membrane that you could color in on a diagram. The blood-brain barrier is the name for the inability or the ability of molecules to get into the cerebrospinal fluid or into the interstitial fluids between neurons. Recall that ependymal cells are neuroglia that make the cerebrospinal fluid by filtering the blood. These ependymal cells exist in the choroid plexuses in the ventricles. These cells, ependymal cells, take blood from a blood vessel and filter it to become the cerebrospinal fluid. Ependymal cells are only one of the neuroglia that filter or participate in this barrier. So this here, right here, is a ventricle. It's actually this one right here. And what we're doing is we're taking things that are in the blood, and these ependymal cells are filtering things to enter into the ventricle. So for example, glucose enters in, and ketone bodies, for better or for worse. But things like red blood cells, white blood cells, no, they're not invited to the party. Astrocytes also maintain the blood-brain barrier, and these were the cells that take substances from blood vessels and feed those substances to neurons. By doing so, astrocytes are like tasked with regulating the interstitial environment of the central nervous system. In the inset picture on the right here, we can see the purple feet of the astrocytes all encircling a blood vessel. Although not neuroglia, and these are astrocytes here, although not neuroglia, the simple squamous epithelial cells that line the blood vessels, and these are those simple squamous cells. And these here are clefts in between the simple squamous cells. And this cell here is called a pericyte, which is just an outer layering or covering. So, although not neuroglia, the simple squamous epithelial cells that line blood vessels have very tight junctions. And this lowers the probability that substances will like leak from the blood capillaries and enter into the interstitial environment. This is a closer up picture of an astrocyte, the purple foot at the bottom, interacting with a blood vessel. And you can see the purple foot and the endothelial cell lining the vessel. There's also blood flowing through this vessel. And as you can see, there are a variety of ways a substance could be transported across the endothelial cells into the interstitial environment. What is allowed to enter are substances that are essential for the brain to function: oxygen and glucose. Nonpolar gases, such as oxygen, are able to enter, but that also means that nonpolar gases like carbon dioxide can enter, right? And carbon dioxide, unfortunately, rapidly changes the pH of the interstitial environment. Other, larger, nonpolar substances like ketone bodies, they can also get in. So can fatty acids. Unfortunately, these things also can lower the pH of the interstitial environment. It's not what we want. What is prevented from entering are any number of toxins or wastes. I can't prevent carbon dioxide from entering, but astrocytes do a really good job at limiting those nitrogen-based waste products like urea that your liver is constantly producing from protein catabolism, right? You're always breaking down proteins. Your liver is always making nitrogenous wastes, and it banks it into urea. So the blood-brain barrier, it prevents waste products like urea, but it also prevents antibiotics from entering because it treats them like toxins. So does your kidney, right? Your kidney and your blood-brain barrier are like, oh my God, what is this? So, and this is why you may have to take an antibiotic like 3 times a day. Your kidney keeps clearing it out in the same way that your blood-brain barrier is like, you can't enter here. So this is problematic for bacterial infections such as meningitis in the central nervous system. Currently, we have few to none that can get across this blood-brain barrier. So good for the brain, but bad if you get infection. But again, this is your blood-brain barrier doing its job. It's saying, I don't know what this is. It must be a toxin. And it's not letting that antibiotic into your CSF or the interstitial environment.